Hi, this is Alex from devstories.eu. AlphaFold Protein Structure Prediction, Episode 7 of 21. Predicting a single protein is like guessing the shape of an isolated puzzle piece. But in biology, proteins rarely act alone. If you throw two random sequences into a standard prediction model, it will often just mash them together in three-dimensional space, regardless of whether they actually bind in nature. To figure out how multiple flexible pieces genuinely snap together, you need a different tool. That is AlphaFold-Multimer. Standard AlphaFold is optimized for single polypeptide chains. AlphaFold-Multimer is a retrained version of the system specifically designed to predict protein complexes. It is trained to recognize and model both homomeric interfaces, where multiple copies of the same protein bind together, and heteromeric interfaces, where entirely different proteins interact. When you provide multiple sequences to AlphaFold-Multimer, it will always return a predicted complex. The critical challenge is determining if that predicted interface is a real biological interaction or an artificial forced collision. The model does what you ask, so it will place the chains next to each other. To solve the problem of false interactions, AlphaFold-Multimer introduces a specialized confidence metric called the Interface Predicted Template Modeling Score, or IPTM. Standard confidence scores evaluate how well a localized piece of a chain folds. The IPTM score entirely ignores the internal structure of the individual proteins. Instead, it measures the accuracy of the relative positions of the chains. It specifically evaluates the confidence in the interface where the proteins meet. Consider a scenario where you're predicting a two-chain antibody-antigen complex. You pass the amino acid sequence for the antibody and the sequence for the target antigen into the model. The system outputs a structure containing both molecules. First, you check the standard local confidence scores for each chain. They might be very high, indicating the model knows exactly how the isolated antibody and the isolated antigen fold. Pay attention to this bit. You can have perfectly folded individual chains with an IPTM score close to 0. If the IPTM is low, the model is telling you that while the shapes are correct, it has no idea how they connect. It merely parked the two folded structures next to each other because you provided two sequences. The interface is meaningless. However, if the IPTM score is high, typically anything above 0.8, the model is confident in the interaction. A high IPTM means the system found structural and evolutionary evidence that these specific surfaces interlock. The model is not just forcing them together, it is predicting a genuine binding event. To rank the final outputs, AlphaFold-Multimer calculates a combined confidence score. This combined metric is heavily weighted toward the interface, typically taking 80% of the IPTM score and adding 20% of the overall structural score. The model with the highest combined score is returned as your top prediction. The structural accuracy of individual chains does not guarantee a valid interaction. The Interface Predicted Template Modeling Score is the defining metric to prove the model discovered a genuine biological complex rather than just fulfilling a request to group two sequences. That's all for this one. Thanks for listening and keep building.